Go. Hi!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker of pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked in research integrity, and I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today it's our news week where we do an overview of papers that we've seen in the news or other sources to try to find an article that we want to cover next week in more detail.
And next week will be our deep dive week, where we take an article that we choose today and look at it figure by figure to learn what it can and cannot tell us. So make sure to subscribe to satisfy your microbiology curiosities.
You can follow along with the papers that we discuss on either week in our shared Zotero library, linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us using the hashtag MicroTWJC hashtag. And boy, do we have a show for you. We're looking about stories about SARS-CoV-2. What is it doing in the brain? Why it is focusing on spike? Why focusing on spike might not tell us all we need to know about why the variants are dangerous. Speaking of variants, how will they respond to vaccines? And how is the evolution of new variants being affected by more people getting vaccinated? Moreover, what could the next treatment for SARS-CoV-2 be? SARS-CoV-2. We'll see some research that investigates. And if you haven't had enough of microbes killing microbes, we have a story about bacteria that predates on meningitis, an evolutionary arms race between cholera and a virus that kills it. And we also talk about what Jurassic Park can teach us about bioengineering. What do I mean? You'll just have to keep watching to find out. But first, I think we have some good news, right?
Yeah, absolutely. Picked out this news story from the World Health Organization that China has been officially certified as malaria-free. And what that entails is that they have to have had like very few endemic cases, like for 3 years, I think. They have like a timeline. And they sort of work with the WHO regulators to identify whether or not they've met those metrics.
Yeah, and I think now they've reported 4 consecutive years of zero indigenous cases, and the thing they've identified a number of reasons for it, because there's some basic healthcare in terms of like mosquito nets, laying down lots of pesticides. So this has been like almost a 50-year kind of program. And recently they've been using this thing called the 137 strategy, where the 1 signifies a 1-day deadline for health facilities to report a malaria diagnosis, and by the end of day 3, health authorities are required to confirm a case and determine the risk of spread, and on the 7 days, taking appropriate measures in that community to prevent further spread of the disease.
Yeah, I think it's first of all, it's great news, right? Because we've talked about malaria before on this channel, and we know that it's a very old disease. It's co-evolved with humans for a really long time, and there was a really big, at one point in history, it was a huge killer, right? And then it went down. It was there was a big push to try to eliminate it from certain areas, and it was successful. Like places like North America and Europe were quite successful in reducing endemic cases. But in other places, especially with more tropical weather, that was a more difficult task. So yeah, I think it's a triumph of not just sort of technological achievements, but also these types of policy interventions, right? Like a public health perspective. Like that's not the only thing that goes into reducing a disease down to 0. And of course, the primary concern now for China will be cases of malaria that come in from out of their country. And if infected people come in and then they don't identify that there is a larger population of I guess malaria travelers, those mosquitoes that are endemic in the country could sample those infected individuals and then put that parasite back into endemic circulation. So I think there's like interesting mirrors, right, when we think about public health in this perspective and the pandemic that we've been going through with SARS-CoV-2, that there are these different phases and different interventions and policy that needs to be deployed in order to control and ensure the health of everybody in a country.
Yeah, I mean that's because what they've done here is it's relatively straightforward. It's what a lot of other countries have been applying. It's just the scale of it. I mean we're thinking back to SARS-CoV-2. I mean, we're comparing that. I mean, malaria doesn't really have a good vaccine for it yet, whereas SARS does. And there are plenty of strategies that have been employed the strategy was quite different from malaria. Things like mosquito nets, pesticide, whereas SARS,
Yes.
it's that social distancing. So it's actually the it's somewhat different, but you can sort of see like there's a through line here of it's not just about having the tools, it's having the strategy to use them.
Yes, yeah, absolutely. Yeah, I think different strategies will certainly be more or easier or more difficult to employ, right? And like you'll have to take different ways in terms of getting compliance with those things. But yeah, it's not, I guess, that's maybe my biggest takeaway from this. Well, first of all, the good news that it can, it's malaria is being fought back in a very large country, but also that, yeah, an important part of public health is not just these like cool technologies, but also the compliance with certain types of rules and ways of ways of doing things.
Yeah, and I think we talk about SARS a lot, but now we're going to talk about directly with a paper that came out on medRxiv, this preprint about SARS-CoV-2 infecting brain astrocytes of COVID-19 patients. So this is like a study that covers quite a lot. So they do some MRI on subjects diagnosed with COVID-19 who've had some mild like kind of symptoms, and they follow up with a very like a questionnaire, and they also do like some autopsies of people to try and they're looking at like how COVID-19 affects people like in living brains with mild symptoms, and they find there is
Yeah.
something that's going on in there in
Yeah. And
yes,
we've heard some of this stuff before, right? Like there was like we talked about the NRP receptor, right? There's a separate
yes.
receptor that is found in neuronal cells. I think that this paper also follows up on that, saying like these astrocytes have that receptor, and that's the way in which SARS gains entry into them.
Yeah, this paper has quite a lot of disparate bits that all come together to build this picture of what's going on in the brain. Because you can't really, if you have someone infected with SARS-CoV-2, you can't really delve into their brain. It's not gonna. If you want to look around, the only way to do it is either through an MRI, but you can't like identify viruses through MRI. So you can see how the brain is changing on that basis, and then once
Right.
you go so the only way you could actually see SARS in the brain would be from an autopsy specimen, but then you can only really get those from people with severe SARS-CoV-2. And then so they take those bits and they melt it together with like some cell culture experiments. So they take like neuron cells, I think brain cells,
Yep.
and yeah,
Hmm.
and then they infect them there, and they kind of link everything together through that. Um,
Yeah. Yeah, their initial observation comes from MRIs, just seeing like that there is this like morphological change in some
yeah,
SARS-CoV-2 infected brains.
Yeah, I mean, in an area of the brain that's like right up near the olfactory bulb, if I'm seeing correctly. So that's kind of
Yeah.
near the ethmoids the area inside the nose essentially that backs up against the brain, and that's
Yep.
where you'd kind of look for if you're expecting damage from SARS-CoV-2. So
Yeah.
yeah, it's quite an interesting paper in looking at that and getting us a better understanding of how SARS-CoV-2 is impacting the brain, and then thinking about how that might affect, like, say, looking at neuronal symptoms or people with long COVID who are still suffering from neurological symptoms.
Yes. Yeah, no, I like it seems like a really interesting paper from that perspective, bringing together like because we've had a lot of disparate little information like in our news programs, we've definitely covered
Yeah,
little pieces of it, but here it's being brought together in this more cohesive story.
yeah, it's something that we'll definitely be keeping an eye on. This is an ongoing news story with what's going on in the brain, and there's more research coming out all the time about it. So
Hmm.
that'll be interesting. Uh,
Cool.
Next paper is re- reduced sensitivity of SARS-CoV-2 variant Delta to antibody neutralization.
Yeah,
Uh,
I'm sure. I mean, we've been talking about mutations a lot, like the variants and how that affects. I mean, the media has been talking about it a lot. So that Delta variant, that's what they're calling the Indian variant,
yep,
or like the one that sort of came to prominence during that particular outbreak. And in this paper, they look at, well, first they look at some monoclonals and how they neutralize these three different strains. And then they look at sera from vaccinated and unvaccinated individuals, both like a few days after vaccination, and then also like after the first vaccination, after the second vaccination. So really straightforward, I guess, sort of work. And it does show that the Delta variant seems to not be neutralized as well with vaccine serum.
Yeah, this is definitely one for the fear column, if I was to put it. So they do
Yeah.
compare like a bunch of strains. So the Delta is doing, but I mean, interestingly, they also test the Beta strain, the South African strain, which
Hmm.
seems to be also be very resistant to antibody neutralization as well.
Yes, but
Which
then
I
I guess
mean,
maybe interestingly, that one people aren't seeing, right, in higher
yeah,
circulation. So, this is we always come back to this point that like antibody neutralization is not the only factor that goes into figuring this sort of stuff out, like whether or not a variant is going to be very successful and sweep through the population. And so, in some ways, this is feeding back into that observation.
I mean, you are absolutely right because when you think about variants, we often focus in on the spike protein, but there could be much more to do it. I mean, there's a whole like DNA cycle, sorry, an RNA cycle that happens when the virus infects a cell, and we have to figure out what's going on in that. And that brings us to the next paper, which
Hmm.
is absolute quantitation of individual SARS-CoV-2 RNA molecules, a new paradigm for infection dynamics and variant differences. So this is a preprint that's just been released, and it's interesting because it's about visualizing the SARS RNA as it causes an infection in the cell. Um,
Hmm.
um,
Yeah, so more than just spike protein, they want to have a method to look at every single type of RNA potentially.
yeah, so they use this technique called fluorescence in situ hybridization, where they have these fluorescent tags to RNA probes, which will
Hmm.
bind to different parts of the SARS-CoV-2 genome. So using this, they're able to use microscopy to actually visualize the location of where the SARS-2 is going in the cell, which is
Yeah.
really interesting. So they end up getting these pictures of like these cells with lots of dots on them. I'm gonna try and pull them up
Yeah, I
on
think they're
this.
all the way at the end because like the preprint style, they just
Yeah,
threw all the
it's
figures at the end of this
a
paper.
very new paper. But yeah, so you can see all these. There are like lots of like little dots that indicate where the virus is, and
Mhm.
yeah,
And those dots, they correspond to specifically like this fish technology. We haven't talked about before. It is something that I think we may have seen like immunohistochemistry where they use probes to look for things. But here, that's essentially what's happening. It's a piece of RNA that is complementary to something in SARS-CoV-2, and then it's also attached to something that glows. So every dot that you're seeing is like specifically that binding event between something from SARS-CoV-2 and something that recognizes it.
yeah, and they also cover the subgenomic RNA as well, which is another. It's like when the SARS-CoV-2 infects, it doesn't just have its whole genome there. It like peels off smaller parts of the genome that go off and do interesting things. And
Yes.
what
Yep.
what really interests me in this paper is it also tries to look at variants. So it
Mm.
compares the B117 variant, which I think is the alpha variant now it's
Yes,
called,
that's right.
and they
Alpha.
compare it with like the original recipe SARS-CoV-2 that came out like in Wuhan, and
Mm.
they find that while they differ a lot in what they're doing inside the cells apparently. So
Hmm.
in this case, it's not just the fact there's a difference in the spike protein. There's a lot of difference in how this virus is behaving inside the cells, and that could be an important thing to consider in figuring out what it's going to be, what this thing's doing.
Yeah, absolutely. I mean, we've talked about it before, and it's good to see that like the focus on the spike in terms of creating the vaccine has produced a measurable difference, right, in the tools that we have at our disposal to deal with SARS-CoV-2. But yeah, the virus uses a lot of other proteins. I mean, we've talked about some of those immunomodulatory proteins we don't know about. We've talked about RNA polymerase and yeah, the all those different all the different things that go into helping the virus replicate in the replication organelles, right? Like proteins that recruit like
Mm.
pieces of membrane and things like that. And when we talk about the variants, yes, it is important to remember that those variants have changes in the spike protein, but they also have changes in other places, and some of those other places just aren't studied. And this would be a way of sort of getting at like seeing the differences there. They can't link them necessarily back to those variants. Those are like follow-up studies, right, that have to go into detail. But here we have a tool that we can look in individual cells and really see those differences.
Yeah,
Yeah, something that
yeah,
stood out to me in this paper was that they could see heterogeneous response in the cells. that, some cells got more infected than others. And that's something that you really only get through microscopy, right? If you're doing if
Right.
you're grinding up cells, you don't see that type of heterogeneity, and that could be an important feature, right, to certain types of infection. But maybe that is the difference between the variants. I mean, we can't say, but having this tool at our disposal gives us more data, more ways of measuring the differences between two viruses.
Yeah, it's like opening up a window into cell and we can see what the virus is doing. We can identify to what genes. It's really exciting. But yeah, speaking of like other things that variants are doing, you sent this really amazing paper by identification of a high frequency intrahost SARS-CoV-2 spike variant with enhanced cytopathic and fusogenic effects.
Yeah. So in this paper, I think an important concept that they're trying to distinguish is that we know mutations happen, right, in SARS-CoV-2, and then some of those get transmitted, and then the ones that are really successful, that's what we see taking over the population. But even within a single individual, there could be a bunch of different mutations that occur, and that results in variants just inside a single person. And those variants could also have different effects. So they end up reanalyzing a lot of high throughput next gen sequencing, right? Within a single individuals and looking for variants that appear over and over again. So these are not variants that are being transmitted between people necessarily, but rather mutations that continue that pop up again in individuals. And they focus down on a specific deletion of I think it's part of the spike protein, though. A deletion of part of the spike protein. But we don't see this. This deletion isn't one that we're seeing being transmitted, but we're seeing it arise in individuals. And they make that spike protein in cells, and they show that it changes or it affects the way that the cells fuse together. I think we've talked about this briefly about syncytia. That like the spike protein, we know that the spike protein helps the virions fuse to cells, but even the spike protein expressed on cells themselves will cause the cells to fuse together.
Yeah, and this is that. I mean, the interesting thing about like that kind of, because we usually think about mutations as for going in between different people, but traveling in between different cells, like things like fusing cells together can really help SARS from like travel faster within an organism, because that cell to cell thing, that's really important. And yeah, it's really fascinating to think about it like that and to see that actually there is this recurrent mutation that happens that is that helps them fuse together, and that commonly comes up in, it's almost like having a mutationally locked virulence factor there, where you've got like something that could harm them if they transmit, but could really help them within the host.
I think that this is a concept I think that has been developing in virology for some time, like the quasi-species idea, that if there is some mutational flexibility, it like sort of by design in a virus, then the virus can take advantage of it within a single host to make sure that it's not just the single entity, right, that is infecting that host, but it's this sort of multiple entity. And the fact that they find this mutation over and over again, I think sort of speaks to that reality that this could be something that is very important for SARS-CoV-2 pathogenesis. It's not just about the SARS you get, but also just the potential for that SARS in your own body to pick up a useful mutation of some sort.
Yeah. So
and this, I just want to say, this spike mutation, they hypothesize it's something about the processing of it. Like it's helping that spike protein become processed and come to the surface of the cell more efficiently. And I guess that would mean then that, this certain pathology of cells fusing together would happen more often.
And the thing I find interesting this is thinking about in the context of the research into variants. A lot of people have been saying that variants tend to come up in people who have had long-term SARS-CoV-2 infections. So this would be interesting for investigating people who have had that long SARS-CoV-2 infection. Also, we've seen some research saying that maybe SARS-CoV-2 establishes a long-term place in the gut and doesn't really transmit out of that. So it'd be interesting to see how this interfaces in that, whether that, if there is a population in the gut that has become kind of benign, then maybe has this kind of
Yeah,
intervariate
has it
mutation.
accumulated a certain number of mutations that like make it not transmissible, but like instead just successful for that new niche that they found, right? Like living in the gut.
Yeah, I mean, interesting the idea of like, I mean, this is going far out, but like, I mean, is it possible SARS-CoV-2 to become domesticated by the body if the
Hmm.
right situations are right? I mean, uh, but going back to variants, I think there is an interesting thing that's happening now that because a year ago we didn't have vaccines and new variants were appearing, but now we've got vaccines. So what does that mean for the appearance of new variants? And that brings us to the next preprint, which is
Hmm.
COVID-19 vaccines dampen genomic diversity of SARS-CoV-2, unvaccinated patients exhibit more antigenic mutational variants. Um, So yeah, we're in this new situation. So they performed like some genome sequencing using 23 vaccine breakthrough COVID-19 patients. So people who
Mm.
had vaccines but then ended up with COVID-19, and they looked at the kind of this. They found that the people tended to have like less COVID-assisted complications, but
Hmm.
also the variation was less than in people who didn't have the vaccination.
Interesting. Yeah, I mean, this this is something that we know we would expect to happen, right? Like, of
Mm,
course, the the vaccine is going to change the way that the SARS-CoV-2 population grows inside of an individual, and antigenic mutational variants. I guess I actually don't quite. Sorry, I'm trying to open the paper. I didn't have it open.
okay. Let's see what they talk about here. Because I mean, usually we think about like genetic drift playing a factor in viral evolution, and now
Hmm.
now we've got these vaccines that are inducing a really significant selection pressure, and
Yep.
the question is like, is that pressure going to like cause more mutants to appear, or is it going to cause it to peter out eventually, or is
So
there going to be yeah.
this is theoretical, or they have
Oh,
the
this is a
what?
sequencing paper. So they've
Hmm.
taken some sequences and then they measure the variants within those sequences, and
Mm,
you
I
take
see.
those to like represent. So at the moment they've got 23 breakthrough through COVID-19 patients that they're comparing to 30 unvaccinated COVID-19 patients at around the same time. Um,
And
and
seeing the types
they
of mutations that occur,
yeah.
yeah, in those sequenced things. Yeah, I
I
see.
mean, there is a theoretical component in there where they're looking at places where there are specific neutralizing epitopes. So they're looking at various studies have now been published that are looking at the
Mm,
specific things that
I see.
our B cells and T cells are reacting to. Um,
Yeah, I think
so
I understand this now. It's like, yeah, what is by picking up this data set, they're answering a question like, what is the pressure that vaccines are applying to different loci on the spike protein?
yeah,
Yeah.
and this like kind of tags into like some of the T cell stuff we looked at a while back, which was looking at
Hmm.
what are the T cells reacting to? Are there specific genes that they react to?
Right. Right.
Um,
And we've seen like prior to this, we've seen this idea that like what are the all of the possible mutations that could happen to the spike protein. But in
mm,
some ways this data is narrowing it down a little bit more because it's saying like, okay, but now we have a vaccine that performs a certain type of pressure. So from all the different mutations that we could see on the surface, right? From like deep saturation mutation, what are the ones that are likely going to come up because of this vaccine? And then that narrows the pool, so then we can ask like whether those things are relevant to disease in any way. I'm not sure if that's how people will follow up on that information, but it's a way that information could be used.
yeah, and also it's interesting the way they chunk the GNR up and basically look at okay which areas are mutating more. So it and
Hmm.
they look at the main epitopes for B cells, and they notice that those areas tend to be mutating more. The places that will be acted on by antibodies. So what I'm presuming is this means the spike protein mostly, and they're finding
Yeah.
that tends to be the area of most mutation compared to let's say the T cell epitopes. So so I'm not sure whether that says that means that like that could be because the B cells are affecting a more are are being the main source of the selection pressure. That the B cells are the main threat against SARS-CoV-2, or that T cells
Hmm.
epitopes are so essential to SARS-CoV-2 that it can't mutate them as well. So it's
Yeah.
uh, so it's an interesting paper to look at from that kind of perspective.
Yeah, totally. I find that very interesting.
Yeah. Oh,
Okay, moving on.
so SARS-CoV-2 mRNA vaccines induce persistent human germinal center responses.
Yeah, so in this one, I guess this is very similar to that idea, right, that we had just left off on, and asking, wh the vaccines are what are the vaccines doing? What does the B cell response to the vaccines look like? And so in this one, they sample people's lymph nodes, and we've talked about this before, that
Mm,
in the lymph nodes is where the B cells are maturing into their final form, like the most evolved antibodies towards the specific antigens they've been tickled with.
mm.
And so here they're actually sampling those B cells and checking out the repertoire and how that changes through vaccination. And so I guess, like, we know this happens. This is the description of it. Just like seeing all those different clones. I picked it up because it's important to think about. We know we've said this before on stream, that like we know the vaccines work from the clinical perspective, right? It is successful in reducing the incidence of disease, but the mechanistic details of that like takes time to unravel. And so here we can see that, oh yeah, like this vaccine is having the B cells react in the way that is appropriate to produce a robust immune response.
Yeah, I'm quite. It's interesting, like how they kind of sample draining lymph nodes, and so they've because in previous studies we've looked at autopsies, but I think this uses like living people, and they take fine needle
Yes.
aspirates of those draining lymph
Yep.
nodes to actually look at the germinal B cell centers, which is quite
Yeah.
a. I
If
know that.
you scroll down to figure two, I don't know if you can see that, they show you like an ultrasound that they did, right? So they're using ultrasound to go in and find the lymph nodes, and then yeah, take a very tiny needle and take out some of that. That A is like is an ultrasound.
Oh wow,
So
yeah,
yeah.
that's pretty fun. That I mean, not I mean it's. But anyway, it's
I'm
pretty
sure it's
cool.
a very difficult technique to pull
Yeah.
off. But as you said, it gives us a sort of a rare glimpse into what's actually happening in vaccinated people. I was hoping, when I originally picked this from the title, I was hoping that we would see more about like what cell types are being transformed by the vaccine, right? Because
Mm.
like that's also part of this story. Like the B cells have to find that antigen first, but it really focuses more on just what the development of the B cells look like. And it looks like it's undergoing something what we would expect, right? Normal somatic hypermutation. Those antibodies are getting better, and then essentially it's a catalog of like all
Right.
the different clones of B cells that they have all these variants, and then which ones go on to mutate and become better.
Yeah, I mean, an interesting thing that came up in this paper, because I think these are people who've just been injected with the vaccine, so that should just have
Yep.
the spike protein. But some of them
Hmm.
have come up with antibodies to other SARS-CoV-2 kind of other
Yeah.
SARS-CoV-2 proteins, and what they link those to is to other coronaviruses.
Hmm.
So they mentioned that those like the epitopes, so the antigens that were reacting to those had more hypermutation, which indicates that those were from memory B cells. So that there might be some crossover in the memory B cell response here that we haven't really seen before. That like kind of the vaccine is waking up,
Hmm.
which is quite interesting. It might be because I mean this because we know vaccine works in a lot of the cases, but also like trying to understand like the little like edge cases of trying to see like what where the variance is coming from. And in this case, if you've had these previous coronavirus that was similar to it, then that could be a source of variance,
Hmm.
and that could be an interesting
Yeah,
thing
and I
to
mean,
think about.
it's something that we might miss as well. I think a later paper we may cover will touch on this idea as well, but like we might miss it if we're always looking for neutralizing antibodies to spike.
Mm.
We may not think about like, oh, like you could also be the immunization with spike may also making neutralizing antibodies to other things, right? And we're just not measuring it or looking for it.
Yeah, that's true. And speaking of other papers, we've got so we've talked about B cells, but now we're going to move on to T cells, which are Also quite important in disease, but we don't. They kill the cells. They kill cells that are infected with virus. And I picked this out because I was quite interested in They have like this new technique where they create these things called spheromers. So
Spheromers.
yeah, they. So okay, right. So this is so. In a lot of studies that study T cells, they have to use a thing that T cells bind to, and T cells they don't bind to just the antigen. They have to bind to this protein called MHC, which presents the antigen, and so you need both of those in order for the T cell to work. But the problem is that the MHC doesn't necessarily bind to its antigen particularly well. It's very non-specific by design. So that's
because you want it's the utility molecule that's always presenting antigens to different proteins. We've talked about, or to T cells. We've talked about this before in the context of epitopes, right? MHC is like processing, is the clamp that holds these epitopes out to T cells.
Yeah. And so what they want to do, so it's good for like the body, but for research, it's not very good because it means that there is some variance in, so there's some kind of question over whether you're measuring is what you're measuring. So you need to create like biochemical tools that can do things better. So that's why these researchers have created this thing called a spheromer, which is this gnarly-looking protein. So
it's these like the MHC molecule with a very specific peptide loaded, the one that you're curious about, right?
Right.
A specific epitope, probably SARS-CoV-2 derived, and then they mash them all together into this sphere.
Yeah, so this kind of molecular machine that is designed to hold these MHC and the epitope you're looking for, and then they pull. So part of this paper is building up. Sorry.
Oh yeah, go ahead. I was gonna say they use these to get the T cells out, right? So T cells will bind to this structure.
Yeah, and so half of this paper is developing that as a model, looking at this new tool, and also half of it is applying this tool, so using it on an ex vivo study of SARS-CoV-2 specific cells, and they show like one of the interesting things they show, just as a demonstration, is that T cells that are predicted to T cells can cross-react with seasonal human coronaviruses, and they're also those T cells that are predicted to cross-react are significantly enriched in COVID-19 patients with mild symptoms compared to those with severe disease. So.
Oh, interesting. Yeah, I mean, this tool, one of the interesting things they point out about this tool is it's not. We've had ways of pulling out T cells before, and it's actually very similar. They use MHC with the peptide on it. But I guess like for some reason, maybe the density of these MHC molecules on the spheromere let the researchers pull out even more of the T cell diversity than they have been able to in the past. So the previous tool was called a tetramer, and it was just like, I guess, 4 of these MHC molecules like bound together. But this spheromere, for whatever reason, they show it in their paper. I see this as like very much about this tool that they're using, and like it's able to pull out even more T cells that recognize this particular peptide. And so it's sort of unclear why that might happen, but that means that they're looking at a greater diversity, and if you can see more detail in the T cells that you can pull out, then you might be able to see more differences.
Yeah, I mean, it's exactly that. It's a tool, and kind of that last kind of finding that they had is more of like a demonstration as a proof of concept. It's not necessarily, I mean, it's an interesting one. I mean, it does raise kind of things like, it might like make people think, okay, well, if we use this tool more, then we might pick out more things like that potentially. So, it's
and not just for SARS-CoV-2, but for any infection, right? Or even other disease states, right? Because like autoimmune, right? This tool is used in immunity. So for autoimmune diseases, people are also very concerned about what T cells are recognizing what epitopes. And there could be things in those diseases that we haven't picked up on because we haven't seen the full diversity of T cells that actually engage that epitope. And so the idea would be that this spheromere might be better at doing that.
Yeah, I mean, so tools like this are quite important to take a look at, just because they could reveal something interesting or just allow a sea change in how we analyze something. So that's why I quite like throwing in one of these papers, just so we can see whether something happens or not. in 5 years we might see a bunch of papers using spheromeres, and we can say, yeah, we were there on the ground floor. Um, and the next paper is taking a bit of a left turn. So what could be better than destroying SARS-CoV-2 with SARS-CoV-2? So
Yes.
yeah, we're talking about defective interfering viruses, which I don't know if you've heard about this concept. They're basically like the freeloaders of the virus world.
Yeah, well, I'm aware that like not all viruses make infectious particles. This is definitely new to me and kind of very exciting to think that you could influence the course of infection by just doping in more of these defective particles.
Yeah. So the idea is that these particles they have like they have like bits of the SARS-CoV-2 genome, the bits of the genome that are kind of related to getting packed into cells and to replicate themselves a little bit, but none of the main things for making capsids. So the idea is that if you inject a cell with RNA of these defective like SARS-CoV-2, they'll outcompete the original SARS-CoV-2. So the SARS-CoV-2, when it's in its final stages, it wants to get packed up into the virion, the little capsids
Yeah.
and little spaceships they use to deliver DNA to each other. But these like kind of defective viruses, their DNA like kind of bats them out of the way and takes over. So
Yeah.
they've got a shorter genome, so that means they replicate much faster. So, and because like they don't have capsid genes, there's still the same number of capsid genes there, and they outcompete the original recipe virus. And so
Yeah,
they
so when one of those defective particles becomes its own virion, and then it goes out and it merges with a cell, right, and infects it, but it doesn't have the machinery to make any more of the capsids that are going to get packaged up, and so essentially it's depleting the reserves, I guess, of these spike proteins and other packaging proteins away from the infectious genome copies.
Yeah, so it's an interesting idea, and also interesting to see it applied to SARS-CoV-2. So they do this is mostly a cell culture experiments, so it's very early stages in using this, and I'm pretty sure there'll be lots of people who'll be very frightened of this idea. So they need to go through extra levels to prove that this is okay. But it's an interesting idea to go for, and they apparently like found that adding these genomes in can reduce like the SARS-CoV-2 infection by approximately half compared to the amount of virus in control infections experiments. So if this is going to be used in treatment,
in cell culture,
yeah, in cell
so it
culture.
has this promising thoughts. Yeah, a very interesting approach that I hadn't thought of.
Yeah, I mean, it's an interesting idea to how do you package this into therapy, and how do you even tell whether it works or not. So it's going to be that'd be an interesting thing if it develops further. Um, and I think that moves us off of the SARS-CoV-2 papers, and now we're going to be going into the rest of what's happening in the rest of the microbiology world.
Or at least what we've seen is happening. What
Yeah,
we've sampled from.
what are the cool papers that we found? Dengue vaccine breakthrough infections reveal properties of neutralizing antibodies linked to protection. So we've been talking a little bit about vaccine breakthroughs in relation to SARS-CoV-2. So it's interesting to see what's been happening for other vaccines and how they're analyzing those breakthrough infections. So there's like this leading dengue vaccine called Dengvaxia, which is a tetravalent vaccine. So I love these brandings for things.
Yeah, the name is just hilarious. It's like the nation of Dengvaxia.
I mean, I can't get over Vaxzevria for for AstraZeneca's Chadox. But yeah, so there are four dengue virus subtypes, and Dengvaxia was basically able to stimulate antibodies against all four of them. Yet the overall vaccine efficacy was really low in children who were dengue negative at vaccination. So effectively, even though it produced really good antibody responses, it didn't necessarily protect people against the disease.
Yes.
So they basically went back to the drawing board here and thought, okay, what is happening with these breakthrough infections? How is dengue virus getting through this? Because theoretically, based on our kind of antibody neutralization data, this shouldn't be happening. And what they kind of found was that the antibodies that are capable of cross-neutralizing the various dengue vaccine variants were good in vitro, but not really protective in vivo. So in a petri dish, they worked fine, but in vivo, it turns out that they didn't have that same effect.
Yep.
So
this is the paper that I was thinking about like a little bit earlier, right? Where like neutralizing antibodies, that particular assay, right, happens in vitro, and it doesn't always correlate to what is going to be protective inside of the human being.
yeah. I mean, so they compared it to wild type, so people who are infected with dengue variant 1 and dengue variant 2, and those were strongly correlated with protection from repeat infections. So you're thinking, okay, well, if that means that immunity to both these will protect most people. So if we have one antigen that covers both of them, it should be protective. But actually, it doesn't necessarily shake out like that all the time. And so this is...
Yeah. Yeah, and it seems like they end up sort of advocating for like more antigen specific type assays instead of broadly neutralizing, because that would give you this understanding of like a different antibody that could be important for fighting off the infection.
I mean, I think another thing that I'd want to bring in here is that this vaccine it wasn't created over the course of a year like SARS-CoV-2. It was created over a much longer time period, but yet it seems it's had a lot less like kind of background like research that's been done on it compared to what's been going on with the SARS-CoV-2 vaccines, where I mean with those vaccines we've seen like T cells, B cell studies, we've had lots of investigations into all the nitty-gritty about how it's... So I mean I think that like when people say that SARS-CoV-2 the vaccine has only been produced in a year, I think they're taking away from like, the amount of work that's been done on each vaccine candidate has actually been massive compared to say other diseases we're doing vaccines for. It's because
I think you were about to say I think dengue does fall into the neglected tropical diseases category.
Yeah, I was about to say neglected. I mean, to be fair, neglected tropical disease. I understand now why it hasn't had that as much. It's in the name. We should not neglect them. We should change the name, rebrand. And the next paper is called Synthetic Auxotrophy Remains Stable After Continuous Evolution in Co-Culture and with Mammalian Cells. And you may not like kind of get this initially from the title, but this paper is about Jurassic Park and why John Hammond just didn't think it through with this park. So I don't know if anyone remembers this from Jurassic Park, because I've memorized pretty much every line of that movie, but the lysine contingency...
Oh, I didn't realize they had built in auxotrophy into the Jurassic Park animals?
Yes, they had. So the idea is that your basic Triceratops, they're wandering around and they've engineered them so that they can't metabolize lysine. So the only way they can get lysine is from their diet. So the idea would be that if we stop giving them it synthetically, then that means that these dinosaurs will drop dead. Because they won't be able to get lysine. Except the problem is like you have to lysine is something quite common in foods. So it's a key amino acid that carnivores can get from meat and herbivores can get through some vegetables. It's just like the amount that's available. And I think the same thing happens with people who are trying to engineer bacteria, where people have been trying to engineer like a lysine contingency for bacteria, where we're growing them in the lab, we're engineering them, but we make sure they can't take in one nutrient. But there are some situations where they can go into an area that has lots of nutrients, and including that one that we've kind of denied their ability to metabolize. So so what this paper is saying is like, okay, this lysine contingency, your park's going to go out of control, dinosaurs are going to take over the world. I mean, I haven't been outside in a long while, but according to Jurassic Park continuity, dinosaurs are out there right now. And so this paper is like, okay, well, that lysine contingency obviously didn't work. So what we're going to do is create a compound that you don't find in nature and make it so that our genetically modified organisms are reliant on that. So that means if they go into nature, they won't be able to find the synthetic compound because it's just not there. So,
and the worry, but the danger with that is that they might find a way to circumvent that dependency.
Exactly, because life finds a way. So,
but in this particular setup, they show that that's actually quite unlikely for for the specific engineering auxotroph that they created here,
Yeah.
right? So I think that that's an important thing to remember. That I'm thinking about this Jurassic Park thing. I didn't realize they said lysine. I'm like, that's a horrible engineer because, as you say, lysine is everywhere. I wouldn't expect
Yeah.
that would successfully control the spread of a dinosaur. But let's say that we did engineer. I mean, in this case, they do engineer is a synthetic amino acid of some sort, right? A reliance on that synthetic amino acid. There's still the worry, right, that given so many generations of these organisms, that they would eventually acquire the ability to use some other substitute maybe for that process, right? Or find some workaround in their genome. And so
Yeah.
the authors of this particular paper take that mutant that was made that requires a specific amino acid, and they put it through an experimental evolution setup, a continuous culture where they're continuously growing up this bacteria, and they're giving less and less of the particular amino acid that's meant to control them. And they find that they never really evolve to stop using it. They evolve ways to use it more efficiently, but never like to completely escape that dependency. And so that's over many, many generations. And I love this picture. This is also so close to my heart because this is a DIY setup. At least it was published a couple years ago. I remember reading about it. All the plans are available, so you can do these types of continuous evolution experiments. And we've talked about continuous evolution before. We've talked about it in the context of engineering ace decoys and escaping and phage therapy, making sure phages, and so essentially this is a type of setup that you can build by yourself that have access to that particular experimental design. And so these authors do that. They evolve their thing. They find that it uses more efficiently, but never fully escapes. And that's good news because it means that control that auxotroph control that they put in is viable. And then they stress test that by co-culturing their bacteria with a cell culture and just seeing that, they still, when you remove the amino acid, the bacteria die in that cell culture.
So their basic assertion is life can't find a way.
And I think it's important to highlight that they chose this very specific, right? It's only true in this one sense that they've illustrated, right? This particular amino acid control in E. coli. And that it's promising because it shows us that this technique is viable, but of course in every different control strategy that you that one could imagine and implement, like you'd have to sort of redo this experiment, right, to validate that it indeed is there are no escapes, because we don't know, right? Like we don't know in evolution like what pathways someone can find in that large network of genetic structure. And what they're showing in this paper is for this particular control, right, there doesn't seem to be a viable pathway for it to escape.
So you make sure you spare no expense when you're trying to develop synthetic auxotrophy and check whether they're stable or not.
Yeah, and I think I just want to throw it out there. This is another use for directed evolution and continuous evolution, right? Is like, it's also not even to make something, but to test whether or not something will remain over several cycles.
Next paper is type 6 secretion system killing by commensal Neisseria is influenced by expression of type 4 pili. So bacteria killing bacteria. Well, is this the theme of this one?
Yeah, bacteria on bacteria violence. And this is, I think we may have alluded to this before, and that's why I picked out this paper. I'm like, I think we talked a little bit, but we've never really presented, I guess, one of the papers from this field of bacterial competition. But bacteria we have talked about secretion systems, right?
Yeah.
Bacteria have secretion systems. That's how they inject proteins into other cells, including other bacteria. And in this particular example, we're talking about a type of Neisseria that colonizes the airways of individuals. Some Neisseria are pathogenic. The one that they're talking about here, I think, is not a pathogenic version.
Nope.
And instead, they show that it can suppress pathogenic Neisseria. And specifically by injecting it using this type 6 secretion system. But the story gets more complicated when they find out that depending on the pili structures, so other structures on the surface of Neisseria, that injection is more or less efficient in being able to kill the prey.
Yeah, pili are these interesting things. So they're like rods they're almost like finger-like rods that live on the surface of bacteria. They're like kind of appendages that they use to like kind of grip to surfaces or to each other. And so and they come to this study with the almost idea that, okay, if bacteria have pili, then surely they'd be more able to resist this type 6 secretion system because they'd be able to like kind of prod them out the way. But what they kind of find is the opposite, where they add these predators, these Neisserias, can't remember the second name, but and they mix them in with these bacteria, these other bacteria, and they find that if these express pili, then actually it makes things worse. The type 6 secretion system is more likely to be able to allow these bacterial predators to attack them. And I think one thing is that,
Oh, I would just say like that's really interesting and useful information because people want to know how to control these systems. And we've talked about this before, like the control of microbiota through competitive mechanisms, right? Like there's this dream that like microbiota are going to be able to, be the new drug source, right? Where we can eat these cells, they can establish themselves in our gut, and they can give us health benefits. But if we don't understand like what those mechanisms are for establishing those populations, then we'll never be able to engineer that. And this type of research is down that line of technology, understanding how to control competition. What are the factors that go into that?
Yeah, and from the applications perspective, one of the main like kind of victims of this Neisseria is another related Neisseria called Neisseria meningitidis, which is related to meningitis. So the idea is, it's another proof of concept thing where you're saying we've got this probiotic kind of bacteria that kills other bacteria. What other bacteria does it kill? Oh, the one that causes meningitis. So that means that this could have some protective effect, and it'd be quite interesting from that perspective.
So that, as always, eLife delivers on the videos.
Yep, always delivering on like videos. I do appreciate the way that they incorporate them into their site. But yeah, aside from the beautiful pictures, now you've got some. This paper is quite interesting as well. It's still eLife. A chimeric nuclease substitutes a phage CRISPR-Cas system to provide sequence-specific immunity against subviral parasites. Now, this is a whopper of a story. I don't know whether you want to take this or not, but this is
I'll give this because I think it also links a little bit to when you were talking about the SARS-CoV-2 battling SARS-CoV-2. A little bit, there's a relation here that in nature there are mobile genetic elements, parts of the genome, that specifically want to get packaged into capsids of other viruses so that they can spread between things, but they do not. So they can spread between cells, but they do not themselves encode any of these capsid proteins. And so in this particular model system, they're talking about a mobile genetic element called PLE, I think.
Yep.
And that genetic element wants to to jump into the ICP bacteriophage to be able to move between cells. And of course, ICP does not want this to happen because it wants its own genome to be packaged inside of its capsid so it can move between cells. So what the authors find here is that ICP has a CRISPR-like system that it has that specifically targets the PLE element to degrade it so it cannot be packaged, which is just wild. Yeah. So CRISPR, not only an immunity system in bacteria to target phage, but also an immunity system inside phage to target parasitic mobile elements.
Yeah, it's a wild story like this because the bacteria it's in is Vibrio cholerae, so that's a important bacteria just from this perspective of we don't like cholera, but also like interesting like because this idea that the virus is now fighting another virus. So these like two rogue genetic elements, and this bacteria is almost using the PLE like selfish gene as a weapon against the virus. and the virus is adapting to that, and it's stealing CRISPR-Cas9, which is a system from another bacteria developed by bacteria to fight against viruses. It's using that to fight. So I believe that they use like kind of it's very a mercenary kind of thing. I think they use like a gun called the gun for hire evolutionary strategy, where like each organism kind of uses these selfish genetic elements to boost its own like kind of survival.
Yes. And I think another interesting thing about the fact that all of this story is taking place in Vibrio cholera is that it's well known that Vibrio cholera has toxins that are associated through that can be induced through phage elements and through mobile genetic elements. And so there's this story could also have implications for human health and for the control of those toxins that Vibrio cholera creates.
Yeah, I mean, if we're looking at phage therapy against Vibrio cholera, I think this is something you need to be aware of just in case it backfires.
For sure, yeah. Another mechanism, another complication, right? A wrench in that therapeutic strategy.
Yeah, because phages are living organisms that are evolving. Well, I mean, we can debate over whether they're alive or not, but the point is that they are molecules that evolve, and that's something that we need to be very careful of occasionally, because they have their own kind of level. I'm not going to say intelligence, but level of kind of evolution that can mess with us if we decide to base treatments around them.
Yeah, and actually, now that I think about it, this also dovetails with when we were talking about Arboretum, right? And the way that phage communicate with each other by some peptide signaling. Yeah. In some ways, that's also, it probably comes from borrowing things from genomes, right? Like phage are borrowing things from different genomes that they infect, and then as selection pressure acts on them, those life cycle. But the reverse happens as well. Sorry, just like this line of thought keeps going. The reverse happens as well, where bacteria borrow things from phage, right, and then end up being used for their own life cycle. And that's that gun for hire thing I think that you were saying.
Yeah. So I think that sums it up. Because this paper is just like another volley in that, where they've discovered yet another thing that these phages have picked up from bacteria to fight against other phages. So it's like a civil war going on. This is like a three-way like kind of standoff. It's I think it's a wild story.
I think the reality, to me, the more stories that come out like this, I think it just underlies how biology is all about, right? These networks of interacting objects that have their own incentive structures and are trying to make it happen in a very specific environment.
Yeah. Oh, I forgot even like the last part of it, where this paper is actually focusing on an endonuclease that the ICP1 has stolen from PLE to fight against PLE.
Yeah, so it dives into the specific mechanism of the CRISPR that's being used there, right? And we know CRISPR has to have a mechanism to degrade its target.
Yeah, so this is really interesting. So now we're moving on to the next paper, because I think this was delving fairly into evolution. The next paper I think goes into a little bit more directly towards that. It's called Gene Erosion Can Lead to Gain-of-Function Alleles That Contribute to Bacterial Fitness.
Yeah, this is just a really strange evolution story, and I think I always like talking about, or I like to see the evolution papers that come from microbiology, because, again, simple systems, right? We have a lot of control over it. We can sequence them rapidly and see exactly what's going on. And this is a really strange finding, I think, where this gene erosion, meaning that the gene is losing functions, right? Actually ends up being adaptive in some scenarios because it contributes to some other not related to the loss of function, right? Gain of function activity. And so maybe I have to actually say the example here. So the example is a specific enzyme, a non-essential enzyme, ends up losing some of its functions in certain bacteria. And so but what that ends up doing is giving those bacteria an ability to resist heat shock. And so they they find this by heat shocking bacteria over and over again and cataloging the different mutations that appear. And they're like, oh, this is a really bizarre mutation that appeared where this enzyme just lost its ability to do something. It doesn't seem to be related at all to the ability to resist heat. But what they track it back to is that that enzyme begins to act as like an early marker of heat damage, so that like at an earlier phase, this enzyme now is defective, breaks apart, triggers the heat response, and then ends up being protective for those bacteria overall, because they've essentially, it turns that that enzyme into a type of sensor, I would say. Yeah, and
it's
oh, go ahead.
Oh no, I was going to say it's like a canary in the coal mine, basically, where this enzyme basically it breaks down much faster, and then all the proteins that are looking around to see whether things are breaking down go, oh no, stuff's breaking down. Let's do a heat shock response, and it ends up making that response go early.
Yeah.
So it's
and so the reason why I find that the reason why the authors find this fascinating, and like I wanted to highlight to people, is this thought about evolution, right? Where you always think evolution, like, we're trying out all these different things and you're seeing what sticks. Well, sometimes some of the things that you try will break other things and ends up solving a problem in a totally different space, right? So like this breaks some enzyme that's ostensibly there to fix a different problem in life, right? And it ends up being very useful in another condition. Just highlighting, I guess, the complicated web of genetic elements and the way natural selection acts on them.
Yeah, I think the subtitle for this paper is "Task Failed Successfully."
Yeah, totally.
That brings us to the next paper called "Global Mapping of Salmonella enterica Host Protein-Protein Interactions During Infections." So this is an interesting one because going back to like bacteria injecting stuff into other things. So Salmonella injects a bunch of proteins into its host cells. So it infects macrophages. So macrophages kind of engulf them, and then the Salmonella basically finds ways to manipulate it. And so what they did here is they effectively they did some protein tagging to see what those proteins are doing inside the cell. So they're looking at what these effects are doing to the host cellular processes, and what does that mean? So they found like
I just want to interject, like for those viewers who have followed our channel, like we've talked about the type 7 secretion system in tuberculosis and how that supports, right, the growth of tuberculosis inside of very specialized intracellular vesicles, essentially hijacking, right, the regular macrophage way of eating. And that's dependent on a whole bunch of different proteins. So Salmonella has a similar life cycle, and we want to, the thrust of this paper would be to understand, like, what are the proteins that go into that process.
Yeah, and the interesting thing they find is that a lot of the proteins that are injected don't just have one function in the cells. They have like multiple targets. So they're not so much targeting an individual protein, they're targeting a whole process. So things
that means
like
highly evolved, right? Like these proteins are like they're jack of all trades. You only have to inject one, and already it's interfacing in multiple systems in the cell.
Yeah, so I mean, they because I feel like I might be getting this wrong, but Salmonella has animal hosts where it has very intense, like, kind of commensal relationships with, where these protein interactions don't lead to disease, but just leads them to live with them fine. But in humans, it becomes worse because we can't react to them in the same way. So the interesting thing here is that they're looking at they attack cholesterol accumulation at the vacuole. So Salmonella gets engulfed by a cell into like a mini stomach that we call a vacuole that's used to digest them, but it modulates that so that it can't get digested, so it can be actually kept safe in there. And they also it also like changes like various like kind of contact points at the like the way the organelle works. So and a lot of this is kind of very traditional microbiology, where they find an effect, then they knock out a whole bunch of genes and check what's going wrong and what doesn't go wrong.
Very traditional microbiology, but again, their original way of getting at it is like this huge omics like approach, where they say like, what are all the things, what are all the different molecules and proteins inside of the macrophages that bind to these Salmonella effectors.
Yeah, so this is they got lots of big stuff happening to put together a network, and then So very traditional microbiology stuff.
Cool.
Next one is reinvestigating the coughing rat model of pertussis to understand Bordetella pertussis pathogenesis.
I mean, this is also classic pathogenesis model stuff, right? This is the characterization of a model, right?
Yeah.
And we've talked about this when SARS-CoV-2 was just starting, right? These were some of the first papers that we were reading about, right? Like, how are people going to study SARS-CoV-2 in animals? How are we going to understand its interaction with immune systems and stuff like that? And this is the model for Bordetella pertussis, or whooping cough.
Hmm.
Apparently, rats can be infected with Bordetella pertussis, and they cough. And so, one other interesting thing that the reason I chose this paper was because I referenced in the past that Bordetella pertussis has a lot of vaccines, and actually it's been observed that the way that the vaccines have changed the different antigens that are present on Bordetella pertussis. So like it's thought that someday we may have like a resurgence of this particular disease, and that's how the authors contextualized their paper as well, saying that this could be a disease that we'll see again. So we want to make sure that we have a robust model so that we can study the effect of different vaccines on this particular bacteria.
Yeah, and this uses a whole body plethysmography, where they put like a mouse in this specialized kind of air sealed cage, and they measure kind of the airflow in and out, as I understand it.
Hmm.
And they use that to measure the amount that these rats are coughing, and they use that so when they cough, there's a specific waveform, because I think because animals, when they cough, they can be quite difficult to spot, especially if you've got very small lungs. Which case, like, I guess you, I mean, I think rats like a lot of their noise is an ultrasound, so it's very hard to pick out sometimes. But so they're using whole body plethysmography to pick out like where those coughing noises, and they found that that rats infected with a certain strain of like pertussis tend to cough
Yeah.
more.
Hmm.
Um, and the great thing about this technique is it's non-invasive. So usually for a lot of animal techniques, the only way to find out whether the disease is in there is through either directly sampling, so culling the animal at specific time points, and and trying to check whether the virus is there. But so
Yeah.
having a non-invasive way of measuring this is really good in terms of reducing the number of animals people have to use in experiments, and it's a bit more humane.
Yeah, absolutely. And but they end up doing a whole battery of tests. Like, this is a very
Yeah.
model characterization. So it's a nice feature that they can measure cough, and that correlates to like severity and like viral load. They also end up looking at some immune cells and things inside of these animals. Yeah, it's very much a model characterization to
Yeah.
show us like, here are the things that you can measure, and here is an example where you can see severe disease versus non-severe disease in these animals.
Yeah, and they have to do that for characterization, because when they have like a new measurement, they have to correlate to most of the old measurements in order to figure out
Hmm.
whether it works or not. So this is very much that kind of a characterization paper.
Yeah.
I am aware that whole body plethysmography has had some controversy in the past, so I guess that's why
Ah,
they're going to go the extra mile to try and demonstrate the how much it correlates.
sure.
Um,
Yeah. And the utility of that measure.
yeah, and I think that
Cool.
brings us to the final paper where we're going to. Okay, I'm gonna try and pronounce stuff in this, but I'm not sure I'm gonna succeed. Okay, but I'm just gonna go.
Give it a shot.
Mangrovivirga, cunically gen nov spa nov, a moderately halophilic bacterium isolated from bioturbated Red Sea mangrove sediment, and proposal of a novel family, Mangrovivergaceae, fam nov.
Yeah. So this paper is in the category of taxonomic description. And so they found a new bacteria, and this happens a lot. Like,
Mm.
we've talked about before in like sort of the sequencing, 16S sequencing. Like, there are tons of bacteria in the world, but and we also touched on this fact that it's difficult work to grow up a
Mm.
unique bacteria, especially one, yeah, especially if it hasn't been grown before and isolated from some environment. So yeah, I love this picture that they have where they take sediment from mangrove forests, and they put little discs full of some decomposing mangrove material, and that's where they are trying to culture this. They want to culture some bacteria from this environment. And so we've seen that technique used before, right? The iChip technique.
Yeah.
What was that? Looking for unique antibiotics, I think. It was.
Yep,
It
for
wasn't. We didn't cover it.
Taxomaxin.
Yeah, we didn't cover it directly necessarily, but like this is like one of the many techniques that's used to culture bacteria that haven't been cultured before. And so they find something new in this environment, and they end up characterizing it to the degree that they can name a new species. So that means placing it in a tree. That means looking at pictures of it under EM.
Yeah.
That also means, oh, they don't have the final one. The other thing they do is they do like a fatty acid analysis. So they like measure all the different fatty acids in the envelope of it, and they show that that's like distinct as well. So they get to name a new bacteria.
Yeah. And what a name! What a great name!
What a name it is.
Ooh.
But like, I think, I picked it out because, well, first of all, I don't think we've ever seen an example of a paper like this. They do come out at some frequency. You would think that in the age of like all this omics stuff, like everyone just wants to characterize everything. But there's still people doing science, like really diving down deep on a single object. And I would assume that the reason they wanted to do this is because they may want to work with this organism in the future to find its properties, whether or not it's like a good probiotic or something like that. Is understanding this bacteria in its environment really important for the ecosystem? Those are some of the things. And again, growing bacteria is one of the first tools of microbiology, right? It gives you access to a whole bunch of other techniques afterwards. So yeah, they figure out how to grow it, and they do some initial characterization.
Yeah, and I think
like what does it use? How does it? Yeah, what does it eat?
Yeah.
These types of things.
Yeah, and that brings us to the end of the news portion of this. And now we have to figure out which of these papers are we going to dive into next week.
Yes. Yeah, what do you got? What are you feeling, Faz?
I am
caught your attention?
feeling the synthetic auxotrophy paper because it's interesting they've got like a whole new amino acid that they're testing, and then there's that continuous evolution. And they do like, because in their supplementary material, they have like a lot of 3D prints of what the things they use for their continuous evolution stuff.
Totally. I mean, that system had come out a few years earlier. This is sort of the system in action, right?
Yeah.
Seeing a lab pick up this tool and deciding to use it. Yeah, I'm down for that. What else interests me here? I mean, the absolute quantitation of individual SARS.
Yes,
It's a pretty monster paper, but that seems very interesting.
Yeah.
And yeah, I mean, those two are the big ones for me. I would definitely say.
Yeah, I feel because sorry.
Brain astrocytes, I think, are also pretty interesting. Just
Yeah.
because we've seen a lot of elements of that story before, and like to bring it all together would be cool.
True. I mean, I think for me, the absolute quantitation is top of my list because it's got lovely pictures that show interesting things. But then again, so does that astrocyte paper.
Yeah.
But I'm leaning towards the kind of the absolute quantitation because what it might tell us about variants. That's the thing that quite interests me. And also, I mean, they're both preprints, so we can revisit them when they get published as well.
That's true, yeah. So there's also the possibility of just seeing them later and just keeping them on a list and picking them afterwards.
Yeah. So
I'll leave it to you. Both, all three are really good choices in my mind. I wouldn't have a problem with any of them.
Yeah, I mean, I feel like synthetic auxotrophy is quite a straightforward paper, but then there's the chimeric nuclease substitutes, the guns for hire paper. I feel like
guns for
hire because that is just a wild story that I want to get into in a bit more detail.
Yes. Okay. I'm down for that.
Yeah.
Sure.
Okay. Guns for hire. Next week we're going to be looking at. Let me pull this up quickly so that everyone can see. So next week we're going to be looking at. A chimeric nuclease substitutes a phage CRISPR-Cas system to provide sequence-specific immunity against subviral parasites. So that
yeah, wild.
Yeah.
We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible that we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I agree. You can also reach out to us over Twitter with the hashtag MicroTWJC. We both believe that peer review is a process which we can all participate in. So I hope you all had a good time listening to us ramble on about microbiology today. And if you think you have something to add or have found something unclear, please let us know in the comments or on Twitter.
Yep, it's been a pleasure chatting with you, Faz.
Same here, Danny.
Tune in next week for more microbiology content.
Bye.